It's a great pleasure to visit, even though it's only virtually today. When I got the invitation, it was still sort of pre-corona or early corona, and I said, oh, that's wonderful. I wasn't in Heidelberg for a while, and really look forward to visiting. But unfortunately, in the current situation, this is still somewhat difficult. And so we're going to do a virtual seminar now, and then I stop by whenever I'm in the area next time. So my research lab really focuses on innovative computational methods for protein structure prediction, drug design, drug discovery, and therapeutic design. And I'm going to cover all of these areas today and give you some examples. As Rebecca pointed out, my laboratory has grown substantially, and so we work on many different topics, but I hope I can give you a little bit of a flavor of the type of research projects that that interest us. Now, the research I show you today is exclusively from my time at Vanderbilt University. I just started a new institute for drug discovery in Leipzig, but it will take a few more months to renovate all the spaces and bring in all the people and start research in Germany. But I very much look forward to this new task. So let's see, is this also advancing somehow? There we go. So want to give just a very brief introduction. Rosetta was mentioned, and most people know Rosetta for protein folding, and I'm going to show a little bit about that later. But most of the protein structure prediction work we care about with Rosetta is not so much de novo structure prediction, but really modeling complex systems with limited experimental data. And I'm going to show some examples. But in a nutshell, what we want to do is we want to have models that are accurate at atomic detail, derived from experimental data that often do not contain that experimental data. So think of an electron microscopy density map or a nuclear magnetic resonance data set. Atomic detail might be absent, and then adding that atomic detail with the computer program is really one of the strengths of Rosetta. The second area we're going to talk about is small molecule drug discovery. Rebecca mentioned I originally developed that part of the Rosetta software, and we continue to develop that. So that includes docking and design of small molecules in protein binding pockets. We couple that for a long time already with ligand-based approaches. So mostly machine learning approaches, artificial intelligence that sort of link chemical structure to biological activity. And I'm going to show you some examples of that. And then lastly, Rosetta is also well known for its protein engineering capabilities, where you can design proteins. And so we use these capabilities mostly for engineering antibodies and also immunogens, so vaccine candidates. And I will show you some of our work in that area. For most of these examples I show you, I will have a couple of slides. One slide that sort of shows the new or novel computational algorithm that we put in place, and then a second slide that sort of applies this algorithm to a biological, biomedical kind of question. So I have obviously little idea about your background and what you're trained in. So feel free to interrupt me and ask questions as I go, as I don't see you. It's very difficult for me to really get a good feeling for how fast I should go, what you guys are most interested in, how much background I need to provide. So don't hesitate at all to ask questions as I go, and I can adjust which slides I show you as I go, so that we make it in whatever 45-50 minutes or so. So we're going to start with a focus on protein structure prediction. And first of all, I wanted to remind everybody of this holy grail in computational structure biology. Given amino acid sequence, predict the tertiary structure of a protein. And we all know that this is a hard problem. It's a difficult problem. Protein with 100 amino acids, each of them having 100 conformations gives 10 to the 200 possible conformations. So this is an insanely large number. When nature folds proteins, there's no time on Earth to sample all of these conformations. Obviously, you also can't write a computer program to sample all of those conformations exhaustively. This is known as Levinthal's paradox, even though he wasn't the very first person to sort of formulate that. And we know that nature has a guide, right? Nature has free energy. So when it folds protein, it doesn't sample all conformations, but it goes down what we call the folding funnel, minimizing the free energy of the system. And so it only needs to sample conformations that are low in free energy. In the computer, we have a terrible problem. Nature's free energy function is actually quite complicated and difficult to compute for systems as large as proteins. So we need to work with pretty crude approximations in order to compute these or approximate those free energies in a short period of time. And so some of our research is coming up with better approximations that are still fast to compute. And then other parts of the research is coming up with sampling algorithms that are particularly smart about sampling likely conformations, likely low energy conformations. This is an old video that I made as a postdoc when we put the first version of Rosetta in place. But just to inspire people a little bit, this is folding a very small protein ubiquitin from an extended chain with the Rosetta algorithm. This is about real time on your PC. It takes about 30 seconds on one core of your PC to do that. It's a Monte Carlo algorithm that basically makes a change to the conformation of the protein, measures the delta in predicted energy. If the energy goes down, we accept it. If it goes up, we use the metropolis criterion to sort of make a decision whether we want to follow that pathway or not. Gray is the crystal structure, and you see that this simulation for this one trajectory in this one specific case approaches something that's reasonably close to the experimental structure for ubiquitin. Now, this is a stochastic process, and we need to run it several thousand, 10,000, 100,000 times to sample the conformational space, depending on the size of the protein and the difficulty of the folding problem. And again, this is like sampling the whole conformational space. Most frequently, our application cases are either restricted. We might have a template, we might know the fold, or we have some experimental data that sort of reduces this conformational space. This sort of de novo folding in the absence of any experimental data is really pretty rare. So we run these calculations on clusters where we can run 1,000 in parallel, and then it's still half a minute to get 1,000 models of ubiquitin. And then you either look at energy or you look at clustering approaches to find particularly wide energetic minima where a lot of models sort of cluster together. And these are the likely solutions for these kind of problems. So, this de novo folding was the original motivation to program Rosetta. Shortly thereafter, we implemented protein design. And then between these two sort of protein structure prediction, protein design, many other modes within the Rosetta software were developed. We heard about small molecule docking in terms of drug discovery, enzyme design, protein-protein interface design, RNA and DNA modes were input. So it really got a pretty substantial beast. There are over 100 laboratories worldwide that develop the software right now. There are over 500 people who have access to the central code base and modify the code and continue to put cool science into it. It's an academic software package. It's free for non-commercial use. And we have, I believe, far over 20,000 non-commercial licenses. And meanwhile, any major pharmaceutical company has licenses of Rosetta and starts to use it. And so it's really a very dynamic community. It's an academic package. So because it's developing so fast and these are all academics, it's not super well documented. It takes some time to learn how to use it, but it is a very powerful tool once you got through that training phase. So let's look at some of the applications, and let me just check here that I keep the time. Okay. So let me show you at least a couple of those applications that my lab in particular is interested in. So we are interested a lot in membrane protein structure because there are still more membrane proteins that are expected to have a new fold where we don't have a template for comparative modeling. And so a few years back, we wrote a mode for membrane protein folding that allows us to integrate limited data. So for membrane protein, for a number of reasons that I won't have time to get into, we have usually sparse limited data sets. If you do nuclear magnetic resonance, electron paramagnetic resonance, cryo-electron microscopy, or even X-ray, you often don't get high resolution atomic detail data. So this algorithm can integrate all of this data or combinations thereof and fold membrane protein structures quite efficiently. And this would be a typical application project. So this is a mitochondrial translocator protein, TSPO. It's sitting in the mitochondrial membrane of humans, and it's enriched in some cancers. So we are interested in that protein for the development of agents for PET imaging. And for that, we need an atomic detail model to start docking small molecules. And there has been, low resolution EM density maps. There have been some templates from bacterial sources that are very different in sequence. There has been an animal structure from the Zweckstetter lab in Göttingen. And we recently just published a paper combining all of that information and coming up with an atomic detail model for this particular protein, and then docked some of those PET imaging agents to understand where they bind, how they interact with the protein, and how to possibly further improve these agents. And so another example that sort of goes in the same direction of limited experimental data in a membrane protein, that's the multi-drug transporter EMRE. It's an E. coli system that's a model system for some of these transporters effluxing cancer therapeutics from cancer cells and therefore conveying resistance. Here, the method development portion is focused on EPR, electron paramagnetic resonance, is the little sister of nuclear magnetic resonance in studying protein structure. So instead of using the nuclear spin, you use an electron spin. So you have unpaired electrons in your protein. You need to introduce those. Most proteins don't have unpaired electrons, so there is a way to couple a chemical with an unpaired electron to a cysteine. If you do that in 2 locations for 2 specific cysteines, you can measure a distance up to 60 angstrom or so long distances. But for every distance you measure, you need to express a dedicated mutant and measure that distance. And then label the next 2 positions with cysteine. So it's quite tedious, so it needs to sparse data sets. And because these spin labels are very floppy, you don't get like 35 angstrom. You get like these distributions that sort of show you a range of allowed distances, representing the flexibility of the spin label and the flexibility of the protein. And so we have been developing algorithms within Rosetta to model these spin labels and thus use EPR distance restraints for structure determination. Then more recently for this protein EMRE, this particular transporter, we looked at structural models. This transporter goes to a number of states depending on protonation, and really there were good models for some of those states, but not for the protonated state itself. And so via these EPR restraints, we were able to develop a model for that particular state that so far was sort of a blind spot in the mechanism of this particular transporter protein. And I'm gonna just show you the method development here, because this goes to something what I said earlier. How do we approximate the free energy rapidly and accurately? So this is one of our method development projects on the energy function. The classical Rosetta energy function, restart that probably by going back and forward. Let's see. The classical Rosetta energy function, it uses van der Waals, and it looks into some desolvation penalty. It has a hydrogen bonding potential, but it uses, for all of this, it uses the positions of the atoms to compute. So for a hydrogen bond, for example, it needs to compute not just the distance, but also the torsion and several dihedral angles to have the right geometry. And it doesn't consider anything like pi-pi or cation-pi interactions at all. It has a salt bridge potential, but it's very low resolution. So we were looking into a way to sort of position the most likely places for electrons onto amino acid side chains, and then use this location of the electron to create a new energy function within Rosetta that can compute, all of these partially covalent interactions using the coordinates of the electrons. So it reduces you only need to measure distances and angle. You don't need to worry about torsions anymore, because all these connections want to be in a line, want to be atom, electron, atom. And so you can put this in Rosetta. It stays very, very fast, but then you get an effect that, in the traditional energy function, this sort of aromatic cluster here would sort of tend to unfold. We see that here, and then in our modified version, it stays nice and packed together. And so now we are using this also to look in particular in protein-drug interaction, because of a lot of the small molecule drugs have sort of less traditional functional groups compared to amino acid side chains. So I guess with that, I will probably go ahead and look at, maybe I tell you a little bit about this. So this is another application of this protein structure prediction theme. Many of you are aware that we have an explosion of experimental protein structures available to us. So this is already a few years back, but you just see that we are close to 200,000 or so experimental structures in the protein data bank, which means we have a substantial number of structures for the human gene set, but we also can build comparative models for many other human proteins. So the coverage is maybe somewhere around 74% or so, depending on some parameters. At the same time, we also have the Human Genome Project and since 2000 or so, and then with all of the next generation sequencing, it's getting much easier to sequence a patient's data and find out which mutation are personal or unique to this patient. So we have been working with a unit at Vanderbilt called the Undiagnosed Disease Network, where they have patients who have a phenotype, they are sick, but they are not diagnosed. And so they sequence these patients' genome, and then they also sequence siblings and parents that are apparently healthy. And we get a list of mutations and coding regions that are unique to the patient and could be responsible for the phenotype. And so we construct for as many of these mutants, that's usually about 50 mutations or so, we construct models for and try to predict whether or not the mutation is likely to disrupt the function of the protein. We do that by, depending on the quality of the model, clustering the mutation of interest with known benign and known disease-causing mutations, looking at functional sites of the protein, known functional sites, known interaction sites with other proteins, are they disrupted or not? And in case of very high resolution structures, we can also look at the overall energy. How likely is it that the protein would misfold based on this particular mutation? Could it be destabilized? Was there a question? No. Okay. And so this, we packaged all of that in an automated pipeline, and maybe I show you a couple of examples of things we find. So this is one case out of, this is not Undiagnosed Disease Network, that's a cancer patient. EGFR is a receptor. It needs to dimerize, and then it signals, and it's linked to cell growth. And in the intracellular side, it has this kinase domain. And when it dimerizes, one kinase domain activates the other, and that's the signal. So we found a number of patients, or my clinical collaborator, Christine Lovely, patient in the cancer center, who had lung cancer, never smoked in his life. And they couldn't find out the reason until they looked at the sequence of EGFR in the cancer cells, and they found that the kinase domain was duplicated. And then we did a study looking at this kinase domain duplication mutant and showed that the linker is not only long enough to position the two kinase domains for one to activate the other, the linker actually has a sequence that facilitates orienting those two kinase domains in that way. And so based on this model and a bunch of cell-based studies and other controls, this patient was then switched to a drug, afatinib, that inhibits this particular kinase. And at least for the first couple of cycles, you see that this lung cancer mass is actually shrinking. Now, unfortunately, these personalized approaches are used these days only once all these sort of approved traditional approaches are already used up. And in this particular case, the patient was already very sick, and after some more cycles, the cancer started to grow again, and unfortunately, this particular patient deceased. But in the future, I think cancer centers are more and more moving to this sort of personalized approach to treating patients' cancer. And here, this computational modeling can help because the cell-based studies tells you whether the drug acts or not, but the computational model gives you sort of the mechanism behind why it would function. And that's an important understanding to make good decisions about treatment strategies here. That's another story in cancer also. Osimertinib is a new inhibitor of the same receptor, EGFR. And osimertinib is cool because it's mutant selective. So one of the problems with these inhibitors are side effects. But osimertinib has a preference to bind to EGFR that is mutated to be activated. So it has some preference to target cancer cells. Now, there are different mutations that can activate EGFR. The two most important one is a deletion mutation, exon 19 deletion, and the other one is a leucine to arginine mutation. And we have shown in the study that for one class of patients, we expect that they're going to develop a resistance to osimertinib via a mutation from G724 to S. The other class of patients we don't think will do that. And this kind of prediction, and then we tested that in cell lines, is important because in the case where you expect resistance, you might treat the patient differently. You might add afatinib to osimertinib to kind of try and prevent the resistance to occur. But you wouldn't want to do that if the resistance is unlikely to develop. And so these kind of sort of personalized studies is something that I find very exciting, to use the computational structure prediction to help understand and make good predictions for personalized treatment of patients. And that's very important, since as you go to personalized medicine, it gets more and more difficult to test your therapy or therapeutic strategy on a large cohort of patients, right? By definition, if it's precision medicine or personalized medicine, the mutations are private to one or a very small set of patients. And so there must be some way to sort of be particularly careful and intelligent about understanding what the functional consequences of these mutations are, to limit the danger that incorrect decisions are made. Okay, so let's go on and talk a little bit about computer-aided drug discovery applications. And here, the idea is to merge ligand-based and structure-based applications. So structure-based application would be, for example, Rosetta or your favorite docking program for small molecules. There are many out there. Rosetta is nice for some applications because it has this flexibility built in. Protein flexibility is a strength of Rosetta, so you can design and dock ligands while having the protein fully flexible backbone and side chains. And at the same time, we also use machine learning algorithms, and they are useful if you have a large database of molecules tested for a particular protein target, for example, from a high-throughput screen. And then we take those molecules, we translate their chemical structure into a series of descriptors of numbers, and train neural networks, support vector machines, cohort maps, decision trees with these data to find the optimal model or the optimal combination of models that distinguish active from inactive molecules. And once we've trained those models, we can use them for screening. We can screen molecular libraries that we can purchase ligands from, or we can also use them for design. We work with a lot of medicinal chemists and make suggestions on what molecules to design. And so here's one of my favorite new projects. This is the game Folded, which some of you might know, which is a computer game for citizen scientists to fold proteins, predict protein structure, or also design proteins. And we are currently working on a project with Boehringer Ingelheim to develop a drug design mode of this particular project. So that's an early version of this. A little bit further now, but I didn't have a new video. But basically, as you design this drug, as you place it in the binding pockets, you get points for making attractive interactions and trying to optimize the shape and all the interactions for this small molecule. And then the way this is implemented right now is that the players design the molecules from fragments using chemical reactions. So we have every molecule they design comes with sort of substrates and a pathway to synthesize it, so that they are relatively straightforward to make. And let me show you, not this one. Let me show you this one. So this one is an interesting one. When you do ligand-based drug discovery, you translate your small molecule into a long vector of numbers that describe shape, charge distribution, and so on and so forth. These numbers are usually invariant to rotation and translation. So that, you know, whatever three-dimensional structure of your small molecule you enter, wherever it's in space and rotate in whatever way, you get the same descriptor set. Usually this is done via distances or angles. Now, distances and angles will not distinguish enantiomers. They're going to have the precisely same descriptor set. But we also know that you know, going from one enantiomer to the other can change biological activity quite substantially. And so one of the questions in this sort of quantitative structure-activity relation field is how to efficiently encode stereochemistry. And there have been a number of suggestions, but it's not trivial because, you know, the chemistry, we do this with R and S, and then we label each stereo center. But for a neural network, R or S is meaningless, right? If you change a substituent from a fluoride to a chloride, suddenly R turns into S, and there's no logic behind that, right? And so the only way the neural network could do is learn it, but if it's just learned by heart, it cannot be transferred to similar compounds. So you need a purely geometric description of this property of stereochemistry. And so you can do that once you include 4 points, so 3 atoms in our case, and the center of mass. And then you can compute these volumes of these tetrahedrons. And these tetrahedrons also come with a sign. Does the center of mass is on one side or the other side of each of these triangles of triplets of atoms. And this tells you something about the shape. This is shaped this way or that way. And you can use that to create a descriptor that is basically sensitive to stereochemistry. So the signs invert once you go from one enantiomer to the other one. And we can use that to train machine learning algorithms. Here's an example from our collaboration with our neuroscience drug discovery group at Vanderbilt. They are interested in allosteric modulators of G protein coupled receptors in the brain. And they want to up or down regulate those receptors, mostly glutamate receptors or muscarinic receptors. Allosteric, because allosteric sites are less conserved than orthosteric sites, so you can better select between different receptor subtypes. And depending which subtype you up or down regulate, you might have indications for, you know, anxiety, schizophrenia, addiction, you name it. And so this was an example looking for negative allosteric modulators. I'm sorry, negative allosteric modulators of mChlor5. And the hit rate in the original high-throughput screen was very low, 0.23%. And none of the initial molecules was sort of a good lead compound for a drug discovery program. So we went ahead and trained a neural network with the data from the high-throughput screen, and then used that neural network to screen molecules that we could, from libraries that we could purchase from, molecules that had a different chemotype, a different fundamental structure that all of our known actives or inactives, but were predicted to be active and had this property that we think they could be good, you know, good lead compounds for drug discovery. And so, you know, 12 of the compounds that we purchased, I think we purchased about 100. 12 of them had the activity that we were interested in. And then 2 of those basically were extremely active in the nanomolar range even. So that was very exciting. And, you know, they are related, they are very similar. We usually buy at least 2 representatives from every class just to make sure we don't have an artifact or lose a good molecule. And they became starting point for met chemistry to optimize their structure and activity. And then we tested those in animal studies, and they confirmed. Ultimately, they still chose a different starting point for moving it ahead into humans. But this was a very nice example for scaffold hopping, where you start with a bunch of scaffolds and you arrive at a structure that has a different scaffold but the same biological activity. And let me see, I explain quickly the method. I just mentioned G protein-coupled receptors. They are a hot area for structure-based drug discovery right now. Until 10 years back, we had hardly any structures. But in the past 10 years, many experimental structures for G protein-coupled receptors have been determined. Humans have 800 of them, many of them for smelling, but then there are 450 that are sort of putative drug targets. Many of them are already targeted by drugs. I think about 30%, 35% of our therapeutics target G protein-coupled receptors. So it's the single most important class of targets. And in the past, drug discovery has always been done high-throughput screen, trial and error, because it was so difficult to predict the structure of a G protein-coupled receptor. But now with this influx of experimental structures, we and others have developed protocols to really build accurate structural models for G protein-coupled receptors, in particular for the extracellular loops that are very flexible and very difficult to predict. And then we can dock the ligands. This is a peptide receptor, and we can develop small molecules. So in this particular case, we looked into Y receptors and developing allosteric modulators for those Y receptors. These could be drugs for obesity, for example. So these receptors are involved in our how hungry we feel, how quickly once we start eating we feel satisfied and stop being hungry. And if we can modulate those receptors, that could be one possible application for that. And so here's a study where we collaborated with Lady Wu's lab. She determined a co-crystal structure of this receptor with antagonist. And then we built a model of the same receptor with the agonist, with the neuropeptide bound, and could then use that model for drug discovery. And maybe here's another example. So that's one of the glutamate receptors. That was the first crystal structure of glutamate receptor. This is glutamate receptor 1, and here we collaborated in constructing the structure of the small molecule in the receptor, and then in particular understanding the structure activity relation. So here we have biological activity on the x-axis, Rosetta predicted binding affinity on the y-axis for a series of ligands derived from this co-crystallized one. And you see that there's a pretty nice correlation in this particular case when you have a high resolution atomic detail model of this receptor. This is still an unsolved problem. Really confidently ranking molecules with software works sometimes. In this case, it works very well, but overall, ranking large libraries of molecules is a difficult thing to do. And that's why this was an interesting study at the time. Okay, so let me use the last few minutes to chat a bit about protein design. So this is the sometimes called inverse protein folding problem. Given a structure of a protein, design a sequence that folds into that structure. And again, this is computationally not a trivial problem. Rosetta, again, uses a Monte Carlo algorithm looking for low energy structure and sequence simultaneously. We see this here for a core of a protein where the sequence identity is changed and the conformation simultaneously. This is top 7. This paper is from 2003. Was the first sort of protein that was artificially designed. At this time, this fold had not been observed in nature. And so Brian Kuhlman in the Baker lab and Gautam Dantas engineered this sequence to fold into that structure. Now, this protein is a rock, it doesn't have a function, but most, what we are interested in is designing protein-protein interfaces, for example, or protein DNA, RNA, small molecule interfaces, so that you can put function into a protein. So let's look at an example. So again, first detour into method development. One of the important problems in protein design is multi-state design. You want to design one protein to bind multiple targets simultaneously. And just to explain why this could be important and what I'm going to show you in the next slide, imagine you want an antibody that neutralizes all strains of the HI virus, right? Then this antibody would need to bind, a panel of surface proteins of HIV confidently. And this is an experiment that doesn't happen in nature. A patient gets infected with one strain that might mutate, but it will not be infected with a representative panel of all possible. And so some humans develop protein neutralizing antibodies that bind to many, many strains, but more or less on accident, not on purpose. But in the computer, we can run this experiment, right? We can enter the structure of a surface protein of HIV and design one antibody to bind them all. But that's not trivial because you have tens of thousands of atoms in many parallel calculations. And so my students, Alex Savvy here, came up with a new algorithm that made this multi-stain design much faster and much more robust in terms of really finding the optimal sequences. By reducing basically the energy barriers in the sequence search. And then Jordan Willis here showed that this algorithm really allows for this multi-state design one antibody to bind multiple targets. And then Jordan went ahead and looked at a particular antibody in HIV. The antibody is down here in blue and green. Blue is the heavy chain. This antibody almost exclusively uses the heavy chain complementary determining region 3 to bind. So this winds in here between these glycans, winds in. And this is a mimic of the HIV surface protein. And it has a beta strand interaction, which buries a lot of energy and also explains to some extent why this antibody is broad, because it knows backbone hydrogen bonds for binding. And the long CDR3 really keeps the rest of the antibody away from the viral protein. And so mutations over here don't really touch that antibody. And so, but this antibody came out of a patient, so we don't know that it has the optimal sequence, right? It was created by chance. And so collaborators of us tested a panel of HI viruses against this antibody, and you see it neutralizes about 40%. So a green number means neutralization. The number itself is just how much of the antibody you need for 50% neutralization. And then we tested a bunch of in silico re-engineered versions of this antibody that we predicted to be more potent and more broad. And so this is the red box is one of the success cases where this antibody is about twice as broad and 4 times as potent. So usually you need only a quarter of the concentration, and it neutralizes quite a few strains that the original material did not neutralize. And so this is important Because if you know hot and potent antibodies that the human immune system is capable of making, then this becomes a blueprint for engineering vaccine candidates. A vaccine candidate would be, a mimic of the binding pocket of this viral protein for this loop that could then be used as an immunogen to elicit these type of antibodies. And the story for HIV is very complex and complicated. Many people work on this, and we won't have time to go into these details, but there is progress made with these kind of strategies. Here we do the second step. After we know that this is a cool port antibody, the question is, do you and I have sort of the precursor antibodies in our body that could be matured into these protein neutralizing antibodies? So for this test, we looked into Red Cross blood donors in Nashville and looked at all of their antibody repertoire, in particular the HCV3s. There were 25 million or so. 25,000 had this length of 30 that we needed. And then we wrote a computer algorithm to search. We can't model them all in Rosetta, it would take too long. So we wrote a quick screen that's looked for the best candidates, and then modeled them with Rosetta. And so we looked at two things: how stable is this loop in this funny conformation, and what's the predicted affinity. And the lower left corner is good. PG9 is this antibody I showed you before. And you see most of the human ones, these are the top 50 out of this 25,000. Some of them are predicted to bind almost as good as PG9, but they are not stable in this weird conformation. And so, but we also don't expect that any of the HIV naive patients has good antibodies against HIV. The question is now, how many mutations do we need to make this into a good antibody? Because when you get vaccinated or infected, that's what the immune system does. It adds mutations to turn these early antibodies into really potent ones. And so we added mutations to that, and lo and behold, in this particular case, I think this could run. There we go. So we let Rosetta redesign those antibodies, and this is a naive antibody with 5 Rosetta mutations starting to neutralize some of those HIV strains. And so, after design, they move down into this general region where PG9 is located. Okay, let me see that we have some time for questions here. So I guess I better stop. We can turn some of those long CDR3s in peptides, stabilize them, and they are still active against, this is an influenza example. And we have some examples where we then use this co-crystal structure of an antibody with an antigen. This is from Ebola, to engineer small proteins that present that this epitope, and then we can inject that in rabbits, for example, and they start making neutralizing antibodies. With that, I would like to thank people in my group, and obviously funding agencies who support research in my laboratory, and I'm happy to take questions. Let me get out of this laser pointer view, and I'm happy to take questions. Thanks for your time.